Throughout history, scientific discovery and invention has largely been a story about men. But women have also changed the world in ways you probably didn't even realise. They work in all fields of science, but these five have saved so many lives I can't wait to tell you about them. Together they have helped unlock the atom, saved millions from malaria, revealed the structure of DNA while discovering that our genes like to jump, and maybe how we'll cure genetic diseases. Our first woman is Marie Curie. At the turn of the 20th century, physics seemed almost complete. The atom was believed to be solid and stable, the final unchanging building block of matter. Physics was a field overwhelmingly dominated by men, confident in the picture they had drawn. But what they didn't know is that a woman was about to redraw it. Marie Curie, born in Poland and working in Paris, discovered that some atoms are not stable at all. They change over time and transform into different atoms. And as they change, they release energy, tiny particles and invisible rays. And the material keeps emitting this energy steadily and predictably until it becomes something more stable. She named this process radioactivity. With the help of her husband Pierre Curie, they discovered two radioactive elements, radium and polonium, and proved that it wasn't a chemical reaction. It was coming from inside the atom itself. It's difficult to overstate what this meant. Her work launched nuclear physics, enabled radiation therapy for cancer, made nuclear medical imaging possible, and opened the door to nuclear energy. Today, roughly half of all cancer patients receive radiation therapy at some stage of treatment. Entire oncology departments trace their existence to the scientific frontier that she crossed. She remains the only person awarded the Nobel Prizes in two different scientific disciplines. But beyond the accolades, her real achievement was conceptual. She revealed that the atom was not inert, it was dynamic. And that insight altered physics permanently. Our next woman scientist is Tu Youyou. In the 1960s, malaria was still one of the deadliest diseases on Earth. Drugs were failing, parasites were adapting, millions were getting sick every year. In 1967, China launched a secret military project to find a new treatment, and Tu Youyou was asked to lead part of it. This was during the Cultural Revolution, not exactly a stable time for research. She turned to the ancient medical texts. Tu and her team screened more than 2,000 traditional remedies. Most of them didn't work, some of them showed promise, but the results were inconsistent. Then she discovered a line in a 4th century text describing an intermittent fever, like malaria. It suggested soaking sweet wormwood in cold water and squeezing out the juice. That detail caught her attention. Previous experiments had boiled the plant. Tu wondered if the heat was destroying the active ingredient. So she changed the extraction method to use cold water. This time it worked. In 1971, her team isolated a compound that rapidly cleared malaria parasites. It became known as artemisinin. Tu even volunteered to test it on herself before the broader trials began. Today, artemisinin-based combination therapies are the global standard. Since 2000, malaria deaths have fallen dramatically, due in large part to these treatments, especially among children. In 2015, Tu Youyou received the Nobel Prize in Physiology or Medicine. She discovered something ancient and isolated the key ingredient, and it turned into one of the most important medicines of the modern world. Number 3 is Rosalind Franklin. By the early 1950s, scientists already knew that DNA existed, that it carried genetic information, and it was found in chromosomes. But what they did not know was what DNA looked like in 3 dimensions, how it stored information, and how it could replicate reliably from one generation to the next. Without knowing its structure, that question couldn't be answered. Then Rosalind Franklin, using X-ray diffraction, produced an image now known as Photo 51. It captured the unmistakable helical structure of DNA. That image revealed DNA's double helix, and with it the secret of how life replicates itself. Once the double helix was understood, a cascade of science followed. Molecular genetics, DNA sequencing, biotechnology, genomic medicine, and mRNA vaccines. The global biotechnology industry, now worth trillions, rests on the structural insight of Rosalind Franklin's work. In 1962, the Nobel Prize for discovering the structure of DNA was awarded to James Watson, Francis Crick, and Maurice Wilkins. Unfortunately, Franklin died in 1958 at the age of 37, and the Nobel Prize is not awarded posthumously. But her DNA image and the precise measurements it produced were central in solving the puzzle. Only later did the scientific community realize how decisive her data had been. Number 4. Barbara McClintock. By the early 1950s, scientists were just beginning to understand DNA's shape. But Barbara McClintock had already been blazing the trail in genetics. Born in 1902, McClintock was a cytogeneticist. That's someone who studied chromosomes under a microscope. In 1931, using pioneering microscopic techniques, she proved that genes are physically located on chromosomes. She also demonstrated that chromosomes exchange pieces of genetic information during reproduction, a process called crossing over. That discovery explained why offspring from the same parents can look different from one another. By carefully breeding maize plants and examining their chromosomes, she began to notice that certain genetic traits weren't staying put. They appeared, disappeared, and reappeared again in later generations. She had discovered what is now called transposable elements, or jumping genes. At the time, her work was dismissed and sometimes ridiculed. Eventually, she grew so discouraged that she stopped publishing her work. Decades later, scientists discovered that jumping genes are not rare curiosities. They're everywhere. In fact, a significant portion of the human genome is made up of these mobile elements. They play roles in evolution, development, and even disease. In 1983, McClintock was awarded the Nobel Prize in Physiology or Medicine for the discovery she had made more than 30 years earlier. Her insight changed how we think about the genome. DNA isn't static, it's dynamic. It rearranges, it adapts, it moves. Now scientists knew two things. DNA had structure and parts of it could move. Now the next step was to edit it. Number 5. Jennifer Doudna. If Rosalind Franklin helped us see DNA clearly, Jennifer Doudna helped us rewrite it. Doudna didn't come from a famous scientific family. As a teenager, she read The Double Helix by James Watson and became fascinated by the drama and mystery of discovery. In the early 2000s, scientists were puzzled by odd repeating patterns in bacterial DNA. They were labelled CRISPR, but no one quite knew what they did. Working with French microbiologist Emmanuel Charpentier, Doudna helped uncover the answer. Bacteria were keeping genetic mugshots of viruses that had attacked them, and using a protein as molecular scissors to cut those viruses apart. It was an immune system in bacteria, but Doudna saw something bigger. If those scissors could be guided to viral DNA, they could be guided to any DNA. In 2012, Doudna and Charpentier showed that CRISPR-Cas9 could be programmed to cut specific genes. Practically overnight, gene editing went from slow and expensive to fast, affordable, and widely accessible. In 2020, they were awarded the Nobel Prize in Chemistry, the first time two women had shared a Nobel in science. Today, CRISPR is being used to treat sickle cell disease, develop cancer therapies, and explore cures for inherited disorders. For the first time in history, we aren't just reading the code of life, we're editing it. For most of history, their names are largely unknown, but their discoveries are woven deeply into your life. Marie Curie showed us that atoms weren't solid and silent. Tu Youyou helped turn a deadly parasite into a treatable disease. Rosalind Franklin revealed the shape of life itself, while Barbara McClintock showed us that genes can jump. And now Jennifer Doudna has given us the power to edit it. 5 women, 5 breakthroughs, and a reminder that the story of science, like the code of life, is richer, deeper, and more inclusive than we were once told. Thanks for watching. I hope you've enjoyed the story of these 5 women that have changed the world. Please like, subscribe, share, and comment. It really helps the channel. Stay curious, and I'll see you in the next episode of How the World Was Changed.